Hello, everyone. We are back at the conference, the CAMILLE Diaries at Art Laboratory Berlin. And my name is Regine Rapp, and we are here in panel C, Modes of Care. And what I try to say right now is that I'm very fascinated and very grateful to have these amazing 5 speakers in my panel today, 3 of whom are artists that are presented at the moment in the exhibition, the CAMILLE Diaries, New Artistic Positions on M/Others and Life and Care. So we are here also, and this is part of the Art Laboratory Berlin's approach of discussing artistic research in the field of hybrid arts. We are here together with artists, anthropologists, and a biologist. And this is really, in a dry-some way, very interdisciplinary. And we are here also to interdisciplinary discuss a new way of dealing with the climate crisis, a new way of dealing with extinction of species from an artistic point of view. And we are very happy to present here in the panel. We have here Sonia Levy, based in London, today with us, the artist with the work For the Love of Coral. We are very happy to welcome here in the panel anthropologist Stefan Helmreich from MIT or Cambridge in the US. We are very happy and want to welcome the artist Nico Clusten from Ontario, Canada, who is present in the exhibition with a piece, Mud Berlin, that was actually produced for the exhibition this summer. And we're very happy to welcome Tarah Rhoda, an artist based in New York and now coming to us from New York. And finally, we are very happy to welcome biologist and Berlin-based scientist India Mansour. We have a quite extensive panel. We have 5 panel speakers with presentations of each 10 minutes. So I dare actually to invite you to first listen to the talks, and I briefly biographically introduce each of the 5. And then I would like to take the dynamic angle to a general panel discussion. And dear audience, I think it was working really well in the previous panels this morning, in the afternoon. Send us questions and comments, and my colleague Natasha will then play to me. The modes of care, how can we approach that? And we have very interesting 3 artistic approaches to it. Maybe I can make us become aware of it again, that we could understand modes of care as a deep engagement with the world. And I quote Puig de la Bella Casa, a vital affective state, an ethical obligation, and a practical labor, end of quote. So she sees the care in 3 dimensions: labor, work, affect, affections, or ethics, politics. Or actually, to go back to a theoretician, philosopher, and biologist, Tarah, that we have a lot of reference points to in the whole project. She considers care as, I quote, caring means becoming subject to the unsettling obligation of curiosity, which requires knowing more at the end of the day than at the beginning, end of quote. So we might actually think, and this is like a little invitation that we go through the talks and the panel today together with all of us, with the idea that we consider care as a vital practice of critique. And we have here 3 brilliant artistic examples of artistic critique. Let me first introduce you to the artist Sonia Levy, who we are absolutely happy to have her work For the Love of Corals in the exhibition here in Berlin. So Sonia Levy follows an artistic practice that shifts between different modes of engagement. And this is an approach that we actually share for many years at Art Laboratory Berlin, the discourse of the more than human worlds. And she is very engaged in reflecting and researching in the arts and different disciplinary fields to have a better understanding of the era which is known as the Anthropocene, but we can also, in Haraway terms, call it the Cthulhucene. She has been exhibited internationally, worldwide, just to name Centre Pompidou à Paris, Musée de la Chasse de la Nature, Musée d'Histoire Naturelle Paris, in the ICA, the Baltic, Obsidian Coast in Bradford-on-Avon, and Goldsmith University in London, just to name a few, if you allow me. So she has interesting keywords, and I think we're going to take this into our discussion later on. I just wanted to name the keywords. Ecologies of becoming with, imagination of consequences, and corals as makers of worlds. And now we have a pre-recorded video where we are lucky to see parts of the video work, which in Berlin we show as a 2-channel installation for the love of corals. And we have a 10-minute pre-recorded video that I'm going to share with you right now.
Behind the scenes, in the basement of the Ornament Museum and Gardens in London, a team of marine biologists and aquarists have initiated Project Coral, a leading endeavor to breed coral ex situ in specially designed tanks. By mirroring the climatic conditions of the Great Barrier Reef, seasonal temperature changes, solar irradiance, and lunar cycles, within custom-built mesocosms, the team has become the first in the world to induce corals to spawn in a laboratory. I began filming at the Ornament in 2017, viewing this coral restoration project as a case study of new paradigm for multi-species living, environmental conservation, and natural history that are emerging in the wake of the Anthropocene. Corals are canaries in the coal mine, their bleaching and demise markers of the devastating effects of climate catastrophe. Project Coral is an acknowledgement of the precarity of coral reef assemblages in a warming ocean, an affirmation of life forms' entanglements in this new climatic epoch. But the project is equally a rediscovery of the sensitivities of our planet and its more than human inhabitants. Corals are intricate, multi-species and multi-scale ecological units. They demonstrate how life forms are not separated from their environment, but on the contrary, by their sheer existence, make and constitute an environment for themselves and others, contributing to adjacent ecosystems with complex and far-reaching effects. Project Coral expands that assemblage to include scientists, aquarists, and a range of other human and non-human actors. For the Love of Corals is a cinematic inquiry and artist film developed over the course of a year, filming the daily labor of the team caring for these endangered beings. The relentless attempt to resuscitate corals from capitalism-induced extinctions. The film follows the life cycle of the marine invertebrates in the laboratory tanks, replicating the seasonality and moon phase of an Australian reef in the basement of the Ornament. This short presentation will discuss some of the research that has informed this film project, together with excerpts from the film. 1. Corals as subversive figures in the natural order. The Ornament Museum and Gardens is an anthropology and natural history museum in South London, which also has an aquarium in its basement. Tea trader Frederick John Ornament, founder of the museum, turned his extensive private collection into a public display in 1901 in a gesture to bring the world to Forest Hill. Public museums were part of the Western Modernist project and their collection gathered by the expansions of empires. Specimens collected from afar and wide were placed inside the ordered and categorized space of the museum, promoting a modern scientific view of the world. Natural history museums are displays of the classifying and ordering impulse in search of the universal laws of nature that animated the early days of modern science. Embedded within the Ornament Victorian-era natural history collection, nature is presented as a domain of mute materialities, governed by mechanistic principles. The systematic exhibits of the Natural History Gallery present species as extracted from their environment, obfuscating the vital codependency existing between life forms. By contrast, the work of Project Coral makes palpable the tangled web of human, non-human life forms and technologies that the corals depend on for their survival. As a model for the concept of holobiont, a term coined by evolutionary biologist Lynn Margulis, corals are tangible world makers, from their symbiotic relation with an algae dwelling inside their flesh to their skeleton built from bodily secretions, which create shelters from which whole ecologies of living and dying are formed, affecting the productivity of the part of the ocean in which they settled. Coral reefs are oases in otherwise barren seascapes, and we all depend directly or indirectly on the health of these holobionts. Their disappearance prefigures hunger and the collapse of the fisheries of many ocean-bordering communities. Moreover, we are just beginning to fathom how this drastic reduction in life might affect ocean chemistry and biogeochemical cycles of the Earth system. Corals demonstrate how life forms terraform. They alter the environment, making it a place capable of supporting life. They disrupt the canonized animal, vegetal, and mineral categories of natural history. They subvert the notion of individuality to convey this complex notion that life forms are always already entangled and folded into each other at a local scale, whilst affecting global processes. The environment is not imposed upon life forms, but on the contrary, life forms make the various components of and for their own environment. I'm interested to see how this figure of the coral and the urgent restoration project being done to try and counteract their extinction might play out in redefining our ways of relating to and understanding nature, a necessary shift from perceiving it as a separated domain to an understanding of the necessity of the enmeshment of life forms. 2. Scientific caring in the ruins. In a strange turn of events, the colonial urge to gather knowledge during the Enlightenment period, the thirst for and collection of millions of specimens, which filled and built the natural history museums, zoological gardens, and aquariums, has resulted in repositories, representative of past and ongoing capitalism-induced extinctions. Natural history museums and collections stand as testimony of the invention of the colonial Western notions of human exceptionalism, the ways in which certain bodies and territories were turned into resources, but have paradoxically become a tribute to the abundance and variations of life forms that once populated the planet, before this new climatic era. I wanted to examine how this architectural context of a museum, which still echoes the Enlightenment value of man mastery over nature, might become a base for a project with the potential to exemplify a collaborative multi-species survival endeavor. Whilst the coral remain in captivity as part of the ornament living collection, the shared attempt to save them established a new and generative assemblage of life-affirming care. Acknowledgement of the damage caused to coral reefs is embodied in the work of the team at Project Coral, from the precision involved in coral IVF and the protocols of raising corals from their embryonic stage to the level of constant supervision that a project's success requires. There is a crossover between the professional, detached space of the scientific lab and the intimacy and responsibility of caring for a living being. The role of the team is shaped by the custodianship of museum conservation and the epistemology of the scientific method, but the scientists and the corals have also become involved in each other's life. This proximity to and involvement with the corals is the sharing of a territorial space, both the familiar working space, but in a cosmopolitical sense too, in that it recognizes earthly commons. The scientists and the corals are entangled in sharing a space for working, living, and world-making, expanding the range of possible worlds in common. In the museum, an ordering impulse has been replaced with a world-making one, and a network of human, non-human lifeform and technologies coalesce to form this critical practice of care. 3. Documenting and practicing entanglement in the Anthropocene. The tidal pull of the moon is strong, considering how it permeates life forms. Yearly mass spawning of corals is prompted by the light of full moon, simultaneously uniting hundreds of different species at night across the ocean. Moonlight, especially that of the full moon, is symbolic of transformative power. Stories and myths involving moon phases are told as ways to see through darkness, in the absence of the illumination of knowledge, or perhaps as a different kind of knowledge. Indeed, as the objective, stable background for knowledge that nature once represented is now gone, philosopher of science Isabel Stengers points out that the question, what do we know has been transformed into the question, what can we know? It's November 2017, the day of the full moon, and I'm invited to join Project Coral for the yearly spawning. Outside, it's a shivery November day in London. Inside, I'm projected into the tropical climate of the lab, its wetness and briny smell. A pervasive white noise is permeating the atmosphere as apparatus are plugged in everywhere and water is pumped, the instrument sustaining this coral intensive care unit. For spawning to occur during the daytime in the United Kingdom, cycles of the laboratory tanks at Project Coral are kept in such a way that their night is our day. Aquarium curator and Project Coral founder Jamie Craggs tell me that the corals are particularly sensitive to disturbance with sunlight around spawning time, since it could upset the egg's release. Corals having evolved to spawn under the cover of night to avoid daylight predators. Therefore, the team is equipped with red light headlamp as they go in and out of a tarpaulin tent set up in front of the tanks. I feel a sense of trepidation at first when I'm given a red headlamp and step inside. I follow Jamie as he is rapidly shining his headlamp over floating containers. That is where the eggs will be collected and attributed to the right individual. Inspecting the corals up close, Jamie expresses that they are near to spawning. He is looking for the pink bulge of the eggs as they appear in the coral's mouth, a visual cue indicating the colony impending spawn. Later that day, the corals start releasing the eggs, and I can just about film the team prudently picking the pinhead-sized bundles, illuminated by the ominous red of their headlamps. What is the role of filmmaking in this new climatic era? And what are the representational challenges involved in such a paradigm shift? The Western colonial fable of nature as a separate domain has collapsed. Nature can no longer be represented out there as a spectacle to be looked at from afar. As artists, shouldn't our task become one of bearing witness to this critical chain of interdependence brought about this globalized state of precarity?
So that was the pre-recorded video recording by Sonia Levy, and we thank her very much. And I'd like to go directly on, and we come back to the questions and the discussion later on after the 5 talks. And our next speaker has been coming already directly in dialogue with Sonia. And we invited Stefan Helmreich, who is an anthropologist, and he was fascinated with the works of Sonia. And this is actually always the ideal moment for fruitful preparation and dialogue, that not only on the day of the conference and the panel, but actually as to unfold, and as actually an intense dialogue has already been developed. And also Stefan was so kind, and together with the help of Sonia, that he also made a pre-recorded video, and that we will look right away. But let me please present Stefan. Stefan Helmreich, he is an anthropologist at MIT, Department for Anthropology, and his main interest is the category of analysis of life. And his interesting, his ideas about corals were actually unfolded in interesting text that already Sonia had actually in the source book, in the theoretical texts which we exhibited behind the installation. And I would like to quote something here for presenting Stefan. And it is an article, How Like a Reef. And I think this is an interesting approach that Stefan is following of corals. And I quote, "I want to suggest that the idea of reading coral represents an opportunity for reconstructing understandings of the cultural and scientific politics of the sea." So talking about macro and micro spheres, and later on we talk about biosphere, I think this is absolutely interesting. So Stefan very much already went into deep research with corals, and was very happy and ready to respond to our invitation, and has already been reflecting on multi-levels, not only the anthropological level, but a philosophical and actually artistic level of how to understand Sonia's piece, For the Love of Corals. So please let me again share my screen with you to show you Stefan's talk.
News from another pandemic. When he was dying of AIDS, filmmaker Derek Jarman made a final film, Blue. As his health failed, his eyesight left him able only to discern shades of blue. His 1993 film consists entirely of a saturated, static, 75-minute shot of international Klein blue, a color conjured in 1957 by French artist Yves Klein, a shade containing a strong dose of ultramarine, so named, beyond the sea, because its source, from the metamorphic rock known as lapis lazuli, came to Europe from Afghanistan, across the Mediterranean Sea. Jarman's choice of the color was complex, more perhaps about the deep sky, in which his voiceover imagines him walking, than about the sea, the blue sky standing for him for, quote, the admirable austerity of the void, unquote, the frightening comfort of disappearing into air. Think of the color blue now in Sonia Levy's 2018 film, For the Love of Corals, like Jarman's, an elegiac piece, a work about caring in and for embodied ruins. At the Horniman Museum in London, where a team of marine biologists seek to induce spawning in aquarium-captive Acropora coral, scrolling sequences of LED lights simulating sun and moon cycles move through a spectrum that, Levy's film reveals, often lingers in the blue. As coral scientist Pauline Kaniewska and colleagues observe in a 2015 paper entitled Signaling Cascades and the Importance of Moonlight in Coral Broadcast Mass Spawning, quote, blue light detection is a key element for coral spawning behavior, unquote. Levy's arresting images of coral under the artificial blue basement moon of Horniman give us blue not as the dim luminosity of death, but blue as a hovering condition of life, reproducible in the lab and perhaps still a force for vitality in the watery world. In Levy's film, blue also arrives in images of mid-19th century cyanotypes made by botanist Anna Atkins, sunprints that Levy consulted in the Horniman archives. Atkins created the images of seaweed by placing the algae on top of paper treated with potassium ferricyanide and ferric ammonium citrate, paper that exposed to the sun wherever the algae was not blocking the light would turn blue when the page was placed in water. Atkins' photograms left ghostly white algal forms, thick, diaphanous, vivid, against a background blue that made it look as though the missing algal subjects had been restored to the ocean from whence they came. Cyanotype blue is often described as Prussian blue, a color first synthesized in Germany in 1706. Prussian blue, as it happens, was employed by Katsushika Hokusai in his 1829 woodblock print, The Great Wave off Kanagawa, and, as art historian Christine Guth writes, quote, materialized the relation between Japan and the world beyond its shores, making the medium part of the message, unquote. The blue of Anna Atkins' cyanotypes, that sun meets potassium ferricyanide and ferric ammonium citrate blue, also records relations, natural and cultural history. Levy employs the writing of artist Anna Ricciardi to think of these photograms as carrying the spectral signatures of now absent, even in some cases, extinct life forms. Cyanotypes have a message for now, Anthropocene blue. Literary theorist Melody Jew suggests that photograms are more than sun writing, more than inscription. For Atkins' work to exist, paper needed to be saturated with chemicals and then saturated in water. To think of this as inscription misses the soaking necessity of water for this genre of signification. Following Jew, saturation may be the semiotic we need to think now about what Ju calls wild blue media. Getting back to the moon then. The Coral Project's blue light is not so much artificial moon writing that brings coral to life, as it is a saturating energy that can be queried for its source. What does its artificiality tell us about the history of experimental scientific imitations of nature? And queried for its force. Can it operate as a scalable power that can be focused on a project of saving reefs in the wild from Anthropocenic overreach? In a paper I once wrote, called How Like a Reef, a riff on the How Like a Leaf book-length interview with Donna Haraway, I wrote of the shifting ways coral had been used as a figure for the preoccupations of those who encountered it and sought to bring it into scientific apprehension. For 19th century colonial mariners, coral was a hindrance, its hard forms a frustrating, but for some, like Darwin, fascinating architecture. In the early 20th century, as Anne Elias shows in Coral Empire, Underwater Oceans, Colonial Tropics, Visual Modernity, new underwater visions of coral as vivid and extravagantly productive emerged, eventually coming to play no small part in their tourist depredation. At mid-20th century, with the emergence of scuba diving, corals came to invite immersive, fleshy, and cautious encounter, opening up visions of coral as polymorphous, even polyamorous, something brilliantly explored by the trans scholar and theorist Ava Hayward in her work on the sticky sexuality of coral in encounter with human hands. Nowadays, as Iris Braverman has shown in Coral Whisperers, and as Malcolm Schick documents in Where Corals Lie, corals have become nodes in global genetic networks under threat from warming and acidifying seas. Sonia Levy asks, quote, "What is the role of filmmaking in this new climatic era?" Unquote. As For the Love of Corals shows us, it is not to follow in the footsteps of John Ernest Williamson in the Bahamas and Frank Hurley in Australia, who in the 1920s, as Elias documents, quote, produced mass-circulated and often highly staged photographs and films that cast coral as industrious, colonizing creatures and the undersea as a virgin, unexplored, and fantastical territory, unquote. For the Love of Corals brings us into the realm of coral gardens in the machine, where scientists' care for coral requires, yes, a kind of staging, trying to reproduce Australian waters in a London basement, but one that constantly keeps context under examination and revision. Context, in its earliest meanings, refers to a weaving together, which, moving one degree of fabric art over, may remind one of Margaret and Christine Wertheim's Crochet Coral Reef, a collective craft project meant to call attention to climate catastrophe. Levy, in For the Love of Corals, weaves together the texts and contexts of Horniman's scientists to a kindred end, though zeroes in on how marine scientists themselves work their weaves, their dizzying zooming out from multi-species assemblages to beleaguered blue planetarity. News from this pandemic. In The Ocean Carries Memories of SARS-CoV-2, members of the Ocean Memory Project point to the centrality of viruses to life in the sea as agents of mixing and nixing. Viruses are at work in coral, too. In Evidence for a Role of Viruses in the Thermal Sensitivity of Coral Photosymbionts, Rachel Ashley Levin and colleagues suggest that warming oceans place coral symbionts like the dinoflagellate Symbiodinium under stress. Free-living mobile Symbiodinium often arrive to coral to become symbiotic algae when corals emit strong blue light. Infected Symbiodinium, becoming unglued from their blue homes, are now experiencing an epidemic emanating from climate change. They are elements in the unhappy imaginary companion film to Levy's For the Love of Corals, a dystopian film that we do not want to see, a film that we must help work to unmake, a film called Unblue.
Thank you, Stefan, so much. And that was really a wonderful production. And thank you to Sonia to produce these two video recordings. As I said, there are a lot of questions. Also, for instance, the filmmaking. We will come back to this in our panel discussions later on. I would like actually already to go on to our next speaker, to Nico Clusten. And this was a really fascinating collaboration in the exhibition CAMILLE Diaries. As actually it has, and Stefan says it at the beginning and the end of his video talk, the pandemic. The challenge of the pandemic for the exhibition project was that we could not have all the artists in Berlin. And Mud Berlin, the project Nicole will also talk today about, is actually then was produced by us, the Art Laboratory Berlin team, by the concepts, ideas, and ground information from Nicole. First of all, Nicole is following her artistic research in the field of the ecosphere microorganisms. It was so refreshing in our talk on the Zoom the other day that I found out that your fascination also for microorganisms started with the becoming aware of the microbiome in and around our bodies. So, see, I can only think about all these aspects, but biographically, I wanted to present Nicole. Nicole is from Burlington, Ontario, in Canada. She just finished her art PhD in visual art at York University in Toronto. What happens when we acknowledge, is her question, through an embodied experience, our connection to a world teeming with life both around and inside us? That's what I just said, to become aware of the multitude of us and not the singular entity. It is also self-understood, and Nicole as well has exhibited worldwide and has now actually been answering, as all artists in the exhibition, to the open call of Mothers and Future Humans in 2018. And since then, we are in contact. And she will elaborate now on the project Modern Berlin, but I guess this is just an example of her artistic approach. I would like to invite Nicole to talk.
Thank you very much. So in my ongoing practice-based research into microbial life, I've been exploring borders, responsible human, non-human collaboration, and our connection to a broader ecology. I work with mud and the microbes that live in it to explore the enmeshed nature of life. As was just mentioned, I was really immediately fascinated, but also repulsed when I became more aware of the human microbiome. And since then, I've been grappling with my relationship to microbes, both around and inside me, as well as webs of connections that extend far beyond my body. In my work, I collect mud and place it along with nutrients that encourage microbial growth in tall sculptural prisms. When exposed to light, the microbes already present begin to flourish to the point that they can be seen with the naked eye. And they form vibrant marbling across the surface of the sculpture. Their visual presence draws attention to their constant invisible presence around, on, and within our bodies. I'm really interested in microbial life's capacity to connect us in a very literal way to each other as well as our environment and other bodies. Microbes really reveal that our boundaries are indistinct and prompt us to think about the many entanglements that enmesh us. Working with microbes has made my current research possible. They've become my thinking partners, guided my research creation, and continue to live within the sculptural work that I helped create. Collaborating with microbes, again, has made my current research possible, but to call our work together collaboration, I think is very aspirational because of the very dramatic power disparity between us. I have chosen to place them within these sculptures, whereas they have a much less active role in choosing to be there. So throughout our collaboration, I have asked, what does it actually mean to form a meaningful relationship with non-human life? And my attempt to reconcile the power disparity between us and continue to try and consider our work together collaboration has been through an understanding of care. So In her book, Matters of Care, Maria Puig de la Bella Casa describes care as, quote, much more than a moral stance. It involves effective, ethical, and hands-on agencies of practical and material consequence, end quote. So care, as it's understood here, is capable of extending ties, of enmeshing or interweaving actors, and caring can be a catalyst for change. But as Bella Casa also stresses, care is not an innocent term. Quote, care in nature cultures cannot be purged from its predicaments. For example, the tendency to pastoral paternalism, the power it gives to caretakers, and the unequal depletion of resources, end quote. She argues that care does not need to be reclaimed from these impurities, but from its oversimplification, whether by idealizing it or denigrating it. And so care between myself and the microbial life in my sculptures is not an act of idealized love or obligation. Instead, care between us is a constantly negotiated and set of constantly negotiated and thoughtful interactions. The task of care really requires me to stay with the trouble by learning to be truly present, to employ two notions proposed by Haraway, in an active engagement that does not turn away from the contradictions, discrepancies, and difficulties present in care, including the difficulty of actually not being present in Berlin to care for the sculptures in this case. So care can take many forms: caring about, caring for, being given care, each with its own characteristics and potential for practical and material consequence. Feminist scholars Joan Tronto and Bernice Fisher, in their influential work on care, distinguish between caring about and taking care of. Quote, whereas caring about someone or something does not necessarily involve any overt action, taking care of implies the responsibility for initiating and maintaining caring activities. Taking care of requires more continuous time spent and more explicit knowledge of the situation than does caring about, end quote. So it's important to me that I care about microbes through contemplating them, empathizing with them, considering them. But importantly, I also typically take care of the artwork as well through interacting with it daily and remaining attuned to the microbial life's needs. I take care of the microbes by providing light, keeping them warm, changing the parafilm that keeps them moist, and adding more water when needed in response to any shifts I notice. Changes in their appearance or behavior are how the microbes and I communicate. Their vibrancy or lack thereof, amount of gas being produced, height of the water, scent is a big one, all provide information that I respond to by taking care of. So over the course of our relationship, and I've been doing work with mud for maybe 5 years now, I've definitely become a better caregiver. So again, Donna Haraway mentions this as well, that the act of caring, you need to constantly negotiate it and become a better caregiver over time through learning from those you're caring for. So, sorry, one moment. So in order for the artwork to be considered a caring relationship for me, I certainly need to, quote, know enough in order to constantly reassess my relationship with microbial life. And so I tend to spend time being with them, both in the act of taking care of them, but also witnessing their growth and communicating with them through, again, those interactions of change. And over time, those microbial colonies really blossom and shift. It is certainly a living sculpture where the microbial life is constantly in an act of both living as well as dying within the artwork. Although it may not be possible to fully understand maybe what it is to be a microbe, this is another aspect of the work where I do want to consider what it means for the microbial life to be placed into these artifacts that I've built. And I think it's very difficult to shed completely our human way of being or our human way of thinking in order to become microbe. But the effort is what I think is an important aspect of the work, that even through the attempt, we can push ourselves towards a different way of knowing or considering the world. And again, this kind of deep contemplation also attunes us to the non-human experience maybe more broadly. I also want to acknowledge that while I'm impacting the microbial life in these sculptures, they are simultaneously impacting me by literally coming into contact with my body, as well as shifting how I understand the world around me. It is also my hope that through a less mediated experience of non-human life, in this case, microbial life, that viewers can actually encounter these microbes in a way that is, again, less mediated, where we aren't necessarily looking at the microbial life through a microscope or through photography, although at the moment we are. But being in the presence of the sculptures, we can actually have that encounter that is less mediated than perhaps some other methods. And that through this, viewers will also be impacted, become more aware of the liveliness of the world around us, and hopefully inspire greater responsibility towards our ecology more broadly. Thank you.
Thank you very much. Thank you very much, Nicole, for your interesting insight in your artistic practice. It's a great example for hybrid arts. And as I said, I'd like to collect the questions for our panel discussion all over later on. We have, if you allow me, I would already move on to our next speaker. Our next speaker is Tarah Rhoda, present in our group exhibition, the CAMILLE Diaries, with the Hourglass, without the H, Ourglass. And it is an amazing visual statement of biochemical processes. And this is why we wanted to also have Tarah here in this panel today with us. And also it reflects different modes of care. Let me introduce Tarah to you. Tarah is an artist from New York, lives and works in New York. She just finished at the School for Visual Arts, and she is actually leading the biolab at the School for Visual Arts in New York. I can only just say in Germany, there is only one, or actually now coming two, biolabs in art academies. I think Germany has to learn still a lot. It's also interesting that, and no wonder actually, not only here in the panel, but with all artists involved in the project, that Tarah is interested in multidisciplinary approach, and biology and biotechnology especially. And let me just say thank you also for letting us taking care of your work. It was similar to Nicole, that we were actually taking care for the Hourglass. But now I'd like to hand over the spotlight to Tarah, please.
Great, thank you very much. And hello, and thank you for the introduction, and good morning from New York. Like Regine mentioned, I run the SVA BioArt Lab, which is the virtual background that you see behind me, but I'm actually quite cozy in my messy Brooklyn studio. I joined this morning for the wonderful talk from Estrida, and I'm glad to be back. So in my talk, I'll be discussing the inspiration as well as the phenomenon behind the Hourglass work. So let me share my screen. The survival and continuance of flesh is bound to the function and fate of flora. Most of the energy that powers our lives comes from plants, whether it's the food we eat or the fossil fuel that is formed hundreds of millions of years ago. We are because plants are. They sip starlight and exhale a livable world. In addition to sustaining animals like us, this transformation of elements connects moments of production and consumption across scales and species, living and non-living. Energy flows, but matter is recycled. From the fixation of carbon to the transpiration of enormous amounts of water redistributed in the atmosphere, plants are essential to the biogeochemical cycles that we depend on. One of my favorite quotes describing this magnitude of planetary care is from Natasha Myers. She says, "As they exhale, they compose the atmosphere. As they decompose, they matter the compost and feed the soil, holding the earth down and the sky up." Hourglass is a tribute to this marvelous and vast alliance, but also a timely warning of our extractive solutions and reductive confusions. Perhaps one of the most remarkable collaborations of the biological world is the correlation between photosynthesis and respiration. In humans, this metabolic exchange occurs as heme, a red pigmented molecule found in blood, circulates oxygen throughout the body's tissue and returns the carbon dioxide to the lungs to be released. In plants, the opposite reaction occurs, in which the taken breath is processed through photosynthesis. In this reaction, chlorophyll, a green pigment found in plant chloroplasts, captures light and harvests its energy to convert carbon dioxide and water into glucose. The resulting synthesis of this simple sugar acts as a source fuel for the photosynthetic organism, but it also generates a byproduct, which accounts for all of the air that we breathe. In addition, atmospheric carbon dioxide is pulled from the air and locked away in a leafy lattice of sequestered carbon, thus reducing the climate threat, which we so desperately need to abate. Human chlorophyll are strikingly similar, even mirroring each other down to the molecular level. You can see here in my sketch that both are composed of the same polyphyrin ring, the only major difference being the center atom, iron in blood and magnesium in plants. These structural similarities not only indicate a duality of function, but suggest ancestral link in the evolution of these molecules. But the parallels don't end there. A particularly astonishing phenomenon is chlorophyll's ability to retain its light absorption property, even when totally isolated from the plant. This extraction can be done by submerging leaves in an ethanol solution, which causes the chlorophyll molecule to diffuse out of the leaf. When this liquid chlorophyll solution is exposed to UV light, the excited molecules must release the generated energy that would ordinarily be transferred to the rest of the plant. This discharge of pent-up energy takes the form of fluorescence, which causes the chlorophyll to astonishingly glow red like blood. The transparent green liquid instantly transforms, appearing thick and opaque, a deep, full-bodied red that is uncanny in its resemblance to the rich fluid that nourishes and sustains us. Hourglass harnesses this phenomenon as a visceral means of framing the vital and fatally exploitive regard humans often have towards plants and ecosystems. A medical bag filled with chlorophyll hangs from an iron frame, slowly dripping from a syringe into a volumetric flask. A bright white spotlight contrasts the transformation of the rich green solution dangling above, while an ultraviolet light illuminates the accumulating red below. Beads of green swell at the tip of the needle and fall into a puddle of red. Drip by drip, the potential of the plant is suspended and exploited to perform its magic. As the drops reverberate on impact, a meditative rhythm coaxes the abstraction of nature on tap. When the reservoir of chlorophyll becomes depleted, the cellulose ghost of the providing leaves emerge, clear and frail, and stripped of their regenerative abilities. The work embodies the form of an hourglass, a device that measures the passage of time through the duration of matter, emptying from one vessel into another. However, this hourglass, without the H, is not a ticking timepiece, but a looming alarm, an aesthetically privileged lure to stopwatch and hold the trouble. It is a slippery countdown to a climate catastrophe, but like most glasses, it oscillates between half empty and half full, depending on one's perspective. It can't simply be flipped over and renewed. While nature should be capable of rebalancing and redistributing its resources, our anthropogenic excess has caused a metabolic rift that has had dire and irreversible consequences. When I began this project, I was contemplating different juxtapositions of flesh and flora, but I was not aware of the phenomenon that causes the chlorophyll to glow red. I was researching the vascular system in hands and different species of leaves. I attempted injecting and forcing plant veins to uptake my own thinned blood. I even experimented with dosing the flaccid stems of limp roses with Viagra and other medications that manipulate the vascular system of humans. My crude attempts at merging the mechanisms between plants and animals were eventually, and perhaps fortunately, upstaged by chlorophyll's innate ability to demonstrate this visceral duality on its own. My first experience witnessing the liquidated plant transform from green to red was absolutely enthralling. It was an intimate moment of both disbelief and assurance. And while I had drafted a few elaborate installations that involved suspending and pumping this miraculous solution through different entangled tubes and vessels, I ultimately decided to let the simplicity of the molecular marvel stand for itself. The IV bag and syringe invoke the interface of the body and commodified ecologies, while the rapid pace of consumption speaks for itself. The act of dripping has always been a recurring aspect of my work, as I find the moment a part pinches off from the whole to be very compelling. I'm particularly interested in the way that liquid transformations hold and move through memory, time, and space, as there's an inherent intimacy and immediacy and the looseness of wetness. Especially fascinating is the psychological shift that occurs when we eject part of our own bodies, as it reveals and reinforces our fundamental illusions of inside and out. These corporeal thresholds speak deeply towards the abstraction and comfort of the contained self. An attempt to suspend that sense of separation of self we so desperately cling to. Many of my projects attempt to distill the mechanisms of these thresholds as means of exploring the essence through their function and relations. I will leave you with a peek at this project in which I recasted samples of my blood, sweat, and tears as a way to memorialize the moment I shed these rich substances that sustain me. You can see them here circling around in a vessel, and they are my actual samples. They've been preserved and casted using a molecular gastronomy technique. So in the spirit of blood, sweat, and tears, and all that cares, I would like to thank everyone at the Art Laboratory Berlin that put so much effort and care, and hopefully not too many tears, into installing the work, one spinach leaf at a time. Thank you.
Thank you very much, Tarah. That was a very, very interesting talk and a very nice survey of your artistic practice and research. And actually, thank you for visually coming and entering the Berlin exhibition at the end of your talk. I guess also talking about colors and blue, and now we have the red and the green, I think we can elaborate on many levels. And I think later on we should probably definitely thinking also about the biochemical relationships and kinships. But actually, if you allow me, Tarah, I will actually go to our fifth speaker and actually come back with questions later on. Our final speaker today of that very intense panel, Modes of Care, is India Mansour. And as we are sitting in different neighborhoods of Berlin, we are actually here in the same city. Hello, India. I'd like to introduce India, who I actually am very glad to have known already for several years. I will explain why. But first, let me say, India Mansour is a biologist. She's a postdoctoral scholar at plant ecology in the Relic Group, the Institute for Biology at the Free University, Berlin. And she's very interested into microbial communities. And she completed her PhD in river science in Germany and the UK. And we actually have known one another for several years. I think it has to do with our last conference, the Non-Human Agents in 2017, where our colleague Joanna Bergman spoke in the panel with Spela Petrich. So that's our conferential entanglements. And actually we have known one another for quite some time now, and I'm having had fruitful in discussions, and I could learn so much because we have created for a couple of years now, we have this DIY hack, the Panke group, and the Panke is the river, which runs not far from our art exhibition gallery, Art Laboratory Berlin. And we are an artist science group, and we want to basically artistically, scientifically hack the river in different workshops and formats. And in various workshops already, we could learn a damn lot from you, India. And so I actually would like to hand over the words to you. Welcome.
Hi, everyone. Thanks so much for inviting me to participate. Regina asked me to speak a little bit about some of my ongoing research, which actually has a lot of conceptual linkages with some of the pieces in the exhibition. Okay, so today I'm going to be talking about Microbial Biospherics, which is an ecological tool that I've just recently started using and developing methods around for investigating ecosystem processes. So first I'll explain what biospheres are. So a biosphere is a closed ecological system. So it's a system that is supporting life. And there is no exchange of matter outside of the bounds of the system, but there is an exchange of energy. So some examples are shown here. The Earth, the largest one that is the largest biosphere that we know, at least so far. There's also the Biosphere 2 project shown in the middle. This was an enormous earth science, earth systems research facility that was constructed in Arizona in the late 1980s. That has a variety of different biomes inside, ranging from coral reefs to savannas. And there are also places for humans to live, as well as for agriculture. And the idea was to try to construct a facility, a closed facility that could support life. And then finally, on the smallest scale, we see to the right an example of an algal microbial closed ecological system. This was some work done decades ago by Folsom, who is using natural aquatic communities in small closed ecological systems, some of which persisted to, there was persistent biological activity for years in these closed systems. So we can see from some of the examples that I just gave that this type of research has been going on for quite some time. And so what are the motivations behind it? Principally in the 20th century, people were doing biospherics research with the goal of creating systems for long-term space exploration or colonization of off-Earth locations. And so the types of systems that were used in this type of research were often large, at least large enough to be supporting plant life. And the idea was to create life support systems that involved the regeneration, reuse, and recycling of matter to provide air, water, and food. However, there are also implications for biospherics research for life on Earth. Although biospheres are much more infrequently used as an ecological tool. Some examples were given by Escobar and Abadi in a review from a few years ago. where they discuss that biospherics research can also provide insight about stability limits and responses to perturbations of our own Earth biosphere. So I'll just quickly talk about what happens inside of a biosphere, and then I'll talk a little bit about the research questions I have and the developments that I'm interested in pursuing when it comes to biospherics research. So this is an extremely simplified representation of a biosphere. You can see that there are some microbial components here, algae, bacteria. There are representations of a few molecules here, water and glucose, and also of some mineral material. And as I said previously, biospheres are energy open. So there can be light energy and heat energy entering the system, and as well as heat loss from the system. However, they're matter closed. So the amount of matter that is in the biosphere at the time when it is initiated and sealed is the total amount of matter that's there. All right, so what happens inside? This may seem trivial, but organisms live, interact both with one another and with their environment, and they die. And death is really important here because this dead biomass or necromass is what's providing matter for future generations of life. So these processes of decomposition and recycling of matter are critical so that life can persist and sustain inside the system. And living microorganisms are important drivers of transformations of matter that are occurring in the system. And so the biological activity, consumption, decomposition, the production of extracellular enzymes, essentially the way that a metabolically diverse group of microorganisms eat and breathe and grow are transforming the matter that's inside. So the transformations and activities that are occurring inside the biosphere system are linked. The organisms in the system are dependent on others to produce what they need. And we just saw this quite nicely explained by Tarah in the previous talk, this connection between photosynthetic activity and respiration and the production of oxygen and the uptake of carbon dioxide, and then on the other hand, the uptake of oxygen and the production of carbon dioxide. And you can see, so this is not a microbial biosphere in this diagram. This is a representation of pools of carbon cycling on Earth. And you can see here that the these large arrows represent photosynthesis and then the production of plant biomass and carbon that is more persistent. So this process in the microbial biosphere would be done by photosynthetic organisms like algae. And then on the other side, microbial respiration and decomposition. So the various biogeochemical processes and cycles that are occurring are linked together by this community of microorganisms that are depending on one another to produce the various components, compounds that they need to breathe or to eat. Another example shown in the lower right here is the nitrogen cycle, which is a variety of microbially driven processes transforming nitrogen-containing compounds. Okay, so now I'll just talk a little bit about my aspirations for using microbial biospherics as an ecological tool. So the kind of great big scientific question that you can pose using microbial biospheric systems is, what are the ecological and evolutionary determinants of long-term system failure or persistence? So here, system failure would be defined as the failure of biological activity to persist, or the failure of life to persist. So what is it exactly that allow life to be ongoing? We can answer some of these ecological and evolutionary questions in a really mechanistic way by using microbial biospheres with model communities. So some things that I'm thinking about right now in terms of the technical development of the systems are building a multi-species assemblage with the potential to persist. So the multi-species assemblage that's there, the biosphere that I showed in the beginning of the presentation from Folsom had a natural assemblage of microorganisms that were already living and interacting together. Also like the microbial communities in Nicole's Winogrodsky columns, those are natural assemblages that are already interacting with one another. In the microbial biospheres, that certainly is one direction to go. I'm currently thinking about building up multi-species assemblages using model organisms that we have a lot of pre-existing knowledge about, so that I know already what traits do they have, what capacities do they have, what potential is going in, what biological potential is going to go into the biosphere. And can I close all of these loops to create a system that might have the potential to persist for a long time? In doing this work, I am now maintaining microbes with various different needs. So for example, I'm culturing algae for the first time. I've previously worked quite a bit with fungi and bacteria, but growing algae now, I need to have a space to grow them where there's light available, for example, and also somewhat controlled so I know what the light conditions are like. So it's requiring me to really learn about these different organisms that I'm working with and what they need, and also to learn what their cycles are and, you know, be going to take care of them at these time points that are relevant for them. Another question is, what signals of life can we measure? So if you recall the definition of a biosphere, it's closed. And so typically if I wanted to find out if there was microbial life in something, I would grab a sample and plate it and see what grows. But I can't sample inside of a biosphere without violating the need for closure. So I need to think about different types of signals that I'll be able to measure without opening the biospheres. So there's a lot of really great microsensor technology. So right now is really a good moment to be thinking about using these tools in this way. Some things could be, for example, measuring partial oxygen pressure has been done by several researchers, or measuring the heat that's produced as a result of biological activity. Of course, in a little biosphere with the microbial community, it's not going to be that much heat, but there are some very sensitive instruments that can be used for that. And now I'll just briefly discuss a little bit of the research questions that I have. So this process of recycling is really important in the microbial biospheres. And one question that I have is, can the community members in the biosphere completely recycle all of their own dead biomass or their necromass? So there are sort of two components to this question. The first is, what kind of necromass are they producing? What does that look like? What is the quality of these different carbon-containing compounds that is left after they die? And then also, what kind of metabolic capacities do they have? And then do those two things match? Do they have the metabolic capacity? And also, are those things physically, spatially close enough to one another that this degradation can occur? Another research question I have is about the persistence of processes versus organisms. So in order for life to persist inside of the biosphere, these various biogeochemical processes, these cycles need to go on. And so in the case of a very simple community, there might only be one species that has the ability to do a particular step in a cycle, to carry out a part of a process. But ultimately, it's actually not that important what the identity of the organisms in the community are, but rather that this total set of all of these processes can continue going on. And there's an interesting way to think about this from a manuscript from Doolittle a few years ago called Darwinizing Gaia, where he was thinking about the Gaia hypothesis and said, how can we put this into a neo-Darwinian context, and what exactly is the relevant unit of of selection. And he proposed in this paper considering biogeochemical processes themselves as the relevant unit of selection. And this question is also interesting because some microbial genetic material is somewhat promiscuous and can move around and jump around between different microbes in various ways. And so it could be interesting to try to push the system a little bit in the direction of having genetic material moving around. And then a final research question that I'm keen to pursue is what does a trajectory to failure look like? So remember, failure is the failure of biological activity to persist in the system. And there are different ways this could happen. One might be if the community members can't completely recycle all of their own necromass, there's going to be a pool of undegradable material that's growing over time. The matter is fixed, the amount of matter inside the biosphere is fixed, so the proportion of matter that is non-living and that cannot re-enter the living pool will increase over time if the recycling process is not particularly efficient. So if that occurs, then we might see a trajectory to failure that looks like this one here. And I'll just end with a quote from Eugene Odom, who's kind of one of the grandfathers of the field of ecology. This quote already is from the late 90s, I believe, and he was already putting out a call to humans to really think about how we, what it is that we're devoting to our care of the earth. Big house is a reference to the Greek roots of the word ecology. He also calls it here the biosphere, and recognizes that this is providing all of our life support needs as humans. Thanks.
Thank you very much, India. And thank you very much, all 5 speakers. That was a big marathon of knowledge sharing. Thank you so very much. Now let's enter actually the discussion round, and we can see all one another. And before I have questions to a few of you in groups, and I'd like to resume a little bit. And actually coming from the 3 artists in our panel, I find this fascinating that all of you take a non-human subjectivity point of view in your artistic research and in the topic that you have chosen for your artworks. It is also a particularly interestingly inductive approach. It is nature in the foreground. It decenters the human from the center of attention. And that we have been busy at Art Laboratory Berlin with these phenomena already for quite some years. Probably have researched our series, Non-Human Subjectivities, Non-Human Agents. So I think this kind of like is a nice all over context. And comparing here an artistic approach, I come here to say, we can also discuss this later on, we have macro, micro, molecular. So I see actually Sonia's work concentrating on corals, which gives us an interesting macro gaze on a macro scale. So concentrating on mesocosms, but actually understanding corals like a whole ecosystem. So it's model for complex ecosystem, whereas Nicole and the Vinogradsky columns, the prisms is an example from the Berlin mud. I see here actually the micro level and microorganisms, and Vinogradsky columns as a model for complex microbial communities. Whereas then in Tarah, I see here the molecular level and the chlorophyll and indirectly the hemoglobin, showing the molecular kinship between animals and plants. I'd like to start with Sonia, and actually Sonia and Stefan. I'd like, as I was saying already, we were very impressed that already the dialogue has started way before the conference. So I'd like actually to open up some questions and aspects to both of you. Maybe we can have here already a little communication, the 3 of us. And maybe we can start elaborating again the modes of care and thinking about for the love of corals. What has struck me, and I think we can all come to the Anthropocene blue, Stefan, that you have elaborated on in your talk. So it is actually interesting enough also that to debate whether the ancients could perceive the blue, or actually they could not see the blue. Or actually I could make also a reference with the blue to another artist we had in the last panel, Mary Magic. She had the blue Agar River. It was Ramazol blue dye. So actually we actually can even enlarge the phenomenon of the blue. And also it represents textile dyes and is remediated by oyster mushrooms. So if Anthropocene blue is about care and remediation, it's not humans, but actually non-humans who do the labor. So it's basically coming back to the idea that Gaia is a very tough bitch. And so thinking about the Anthropocene blue, and I'd like to take Sonia and Stefan into discussion. How can I see this on? How here I see you all now. This is wonderful. I'd like to roll the ball back to you, Sonia, to think about the mind-blowing work For the Love of Corals to the idea of filmmaking. And if you allow me even one more aspect, Sonia, I have to say this here. We have discussed this earlier in the conversation. What struck me really most is you used the filmmaking, and I'm coming from art history, and I wanted to also reflect about artistic and formal aesthetic qualities that you presented us, if I can phrase it like that, an absolute interesting ontological, epistemological platform to understand the corals by actually offering us a 2-channel video installation. And there I see the juxtaposition between the Natural History Museum, as you said, as the stiff, antagonistic institution coming actually from a divide of nature and culture, putting Western hegemonial systems, colonial systems in the center, and this was above and is above in the 2-channel installation, whereas below, there is then the care for corals. There are the scientists in the cellar, actually, thinking about architecture as a system. I'm speaking already too much, so I actually would like to roll the ball over to you, Sonia and Stefan, together.
Okay, maybe I'll start. Thank you so much for your comment, and to everyone for a brilliant presentation. I have a lot of also question, and it was a lot of information, but I also have lots of thoughts about, what everyone presented. But I will try to go back to my film, and in terms of filmmaking and what you pointed in terms of those 2 screens, and what was important for me, and is probably more visible in that 2-screen installation than in the 1-channel version, that extract that I just that you've just seen, is this idea of the context. And I think Stefan also brilliantly pointed out in his presentation the context in its earliest meaning as a weaving together. So I always try and portray together the coral, the scientists, the instruments in their context, and also the context of the Natural History Museum. And so something that I wanted to say is that, that we represent the corals not abstracted from things. So also weaving together myself, the coral, the technology within the film, not pretend to an objective outside view from above point of view. So folded into their context. And I think one thing that also might be relating to some of the other presentation, and that I try to do in my filmmaking, is the idea of the planetary context, the scale of the folding in the biogeochemical cycle of the Earth. To represent that somehow within the project as well. So move away from the extraction, sort of extractor imaginary then that I inherit, while still pointing at it through, of course, the natural history galleries and all the submerged and hidden stories of colonial powers. Yeah, and not to weave myself out of the film as well. And so I think that was done also through some of the handheld and close-up, where I choose in a way that my presence can be felt, and my emotion and my ponderings. Yeah.
I can free associate some thoughts. I think, yeah, I was really struck in this panel by the various colors. You know, there's the blues and the reds, and therefore, you know, thinking about color and light as simultaneously about materiality, whether that's the materiality of minerals or of film, and also as about semiosis, meaning making, and then also phenomenology, the experience of sensing, whether that's human sensing through things like sight, or chlorophyll animated sensing from plants, or microbial sensing, that color and light seem to kind of swarm together all of these things, the material, semiotic, and phenomenological, and that there's a politics to that. There's a politics of light, or a Gaia politics, or a biopolitics, or a geopolitics, or a sea politics of light that seems to suffuse all of these things that we're all trying to get at. And it made me think of maybe the possibility of putting into this conversation a concept like luminopolitics. I'll type that in the chat. And one data point for luminopolitics comes in a paper by a media theorist called Pansy Duncan, thinking about just the politics of color in light in film. And here we've got, with Sonia's work, this kind of amazing work with the color of the film and the color of the LED lights and the colors to which the coral respond. So just thinking about part of what seems to be at stake here for so many of us is care that involves the care of light, right? And I think that that's what some of the people in the Horniman basement are doing, right? Sort of like trying to enact care through lighting. So that's sort of an initial thought.
That's wonderful. We absolutely will pick that up, Stefan. Thank you so much. I will roll over the ball over to Tarah. So that is actually the inviting moment. Thinking about colors and the biochemical processes that you have described. I was struck by the apparatuses that you actually show us in your work, Tarah, in the Hourglass. And I'm thinking also about the interaction that Karin Barat suggests us to be entangled, to be in the matter, to be bodily involved in understanding the matter. And I was struck already in the preparation of installing the Hourglass in Berlin, but then also now with your explanation, and understanding a molecular level of sorts is always abstract. But then to make it visual, comprehensible, it's not only the color, and actually Stefan was talking about the blue and the red. Now we have the green, which is actually then a complementary contrast, the red and the green. Actually, can you elaborate a little bit more about the apparatus and why you chose the IV bag and the syringe? It has a little bit of a a medical connotation, and the needle has also something as if starting an operation of sorts. So it is a process that you show, and then the dripping is actually another, actually also a dramatic moment, referring us to time. And as Stefan said, when we wanted to reflect luminopolitics, actually we have the phenomenon, the temporal category here coming in. So this is a little bit how I wanted to roll the ball over to you, Tarah, if you wanted to reflect or elaborate a little bit on that.
Certainly. The apparatus itself, in terms of the form and the icons that it borrowed, were definitely a way of invoking the body and invoking that that very fleshy, alarming sense of threshold. You know, a needle sort of dangling and dripping immediately brings up you know, notions of being injected or that the crossing of that threshold. And I think that one of the things that's interesting about the project and a lot of the ways in which I approach trying to understand these thresholds is that ultimately I end up needing to sort of extract and excite the matter that I want to reflect on. And even if I'm critiquing the reductive qualities or regard to it, ultimately that's what I end up doing to understand it better, you know, to restage it. So for example, with the photosynthesis and respiration, in order to speak on the metaphorical duality, I had to extract and excite the physical duality in a way that is quite reductive and oversimplified. But that's something that I keep coming back to. And I think that there's something rich about that. And you mentioned, I think you said, understanding in a bodily involved way. And that's definitely where the apparatus and the needle and the urgency of the drip come in. But it was also something that came up earlier when I was listening to Estrida's talk. It's not unusual for me to be incredibly jarred by her words. And by jarred, I mean moved out of my container and a displacement that I want to preserve, you know. But in this scenario, she took me off guard, and I was struck by her raw, live thoughts. And as I listened, I began peeking up these goosebumps on my arms, and the surface of my skin was, you know, increasing in its grip. And so I was thinking a lot about the physical experience of these intimate biochemical reactions. And for me, goosebumps are sort of the essence of care. You know, they're the body being flooded by a response of overwhelming care, whether that's fear or admiration or joy. So that's kind of where my head was today in terms of deduction and simplifying these things.
Fantastic, Tarah. And I'd actually like to roll over the ball to Nicole and India together, because you also had already dialogues before the conference started, and I like this kind of communicative entanglements. And I remember that you found common grounds, and I think this refers to Sonia's work and to Tarah's work and to what Stefan has also reflected, the common grounds on the phenomenon of the microorganism and microorganic planetary entanglements, and how the temporal category kicks in and also the light kicks in. And if you allow me to say that, from a curatorial perspective of care in Berlin, whenever we don't have opening hours, we are putting actually the light in front of the Gradsky columns. What is it all together? It's the red and the blue. Ultraviolet and the LED, 19 hours a day. So this is actually just to tell you of the off the public exhibition time, it's our form of care. But maybe I roll over the ball to Nicole and India.
Sure. I mean, I can start if you want. Yeah, certainly I think those acts of care of the light is incredibly essential to the growth of the microbes. And it's probably the most important element in terms of caring for them in an active way, that we are lighting them, because as you kind of mentioned, they do have their own relations and community within the sculpture where they are also caring for each other to a certain extent. I was particularly interested, and we discussed this on our Zoom chat, in the fact that the microbial colonies in the columns don't reduce in diversity over time very much. That the communities of microbes actually, because they need the waste of another microbe and the byproduct of each other in order to actually stay alive, that they support a community. So certainly symbiogenesis is much more present, I would say, in the columns than maybe competition, which I think is kind of a hopeful, maybe naive way of kind of viewing microbial relations, but also just that idea of constant negotiation of a community within a contained sort of system.
Yeah, well, I'm sure the prevalence of these processes are also changing a lot with time as well. Like what's occurring when there's this big initial disturbance and the mud is first taken and then put into this column, and the environmental conditions are really shifting, and then you have the buildup of this oxygen gradient, and yeah, you kind of have this like this period of the forming of the world inside of the Winogrodsky column, which I'm sure there's yeah, a lot of competition, but also like stress response in response to this perturbance. But then, yeah, like as it becomes a more stable system and you have these beautiful gradations of the different microorganisms, yeah, changing over this time period. Yeah, it was really striking. I was really very glad to be able to see your work in person and hang out and yeah, look at the microbes. Regina and I actually were sitting on the floor next to them, having a conversation and checking them out. Yeah.
I guess thank you, India, Nicole, Tarah, Stefan, and Sonia. My next point. And we actually have more time than 10 to 6, just for the audience to know. And I actually saw already the 3 audience questions, but I keep them a little bit aside because I would like to have another round with my speakers here in the panel. I said I would like to elaborate also specific methodologies. And maybe we have touched this, but maybe to make a few more comments from your side. I'm struck, especially with the choice of you as 5 speakers in that panel, how, and I exaggerate intentionally, how we can easily compare scientific and artistic methodologies here together. Measuring, filming, recording, viewing, embodied experience. Is it a collection of embodied experience? Is maybe the difference of the art and the science, what do we do with the results? Or is the difference the aesthetics, which is more or less present? I'm just putting heretic questions, of course. So maybe who wants to actually reflect quickly on that? Later on, I have another topic I'd like to come to that.
I mean, just briefly, the first thing that kind of comes to mind is Natasha Myers, who talks about protein modelers in her book, Life is Molecular, and really, or Rendering Life Molecular, sorry. And really that an embodied kind of understanding of these particular scientists is really essential to their work. And that maybe we kind of like abstract away from that when we're reading a scientific paper, but the kind of embodied reality of doing science is really much more related to like making than maybe we often assume.
Another comment from someone here about comparing artistic and scientific methodologies.
Well, you also mentioned the term model as applied to our art science practices. And I think that's curious because in science, the term model means to make simpler for means of understanding one aspect of a very complex system. And the goal is to assume as little as possible and account for as much as possible. But at the interface of art and science, for me anyway, accuracy is more aligned with how vividly or poetically or impactfully my metaphor lands. So when that comes to modeling, I think that's where some of the extraction becomes very important for me, and becomes something that almost feels essential, like something I can't get away from, even if that's what I'm critiquing itself.
Thank you, Tarah. I see Sonia has a question to India.
Yes, thank you. Yeah, I was really really intrigued by this idea of Darwinizing Gaia. That you said Gaia, to put Gaia into a neo-Darwinian context. And for me, that's quite antithetical, like quite paradoxical. So I was wondering if you could elaborate on that a little bit, because it's a little bit kind of paradox that I also was trying to portray in my film. So yeah, about two different worldview basically. And so yeah. Thank you.
Yeah, well, I mean, I think the Gaia hypothesis is, always been somewhat controversial in the world of science, because it was also published, by Lovelock, meant to be read by a layperson, but then it's quite, taking a lot of these scientific ideas and like the development of the Earth as a place that is now supporting life, that is sustaining over time and persisting. And there've, been a variety of different scientific critiques. And I think that what Doolittle was trying to do in 2017 was saying, if we shift the way that we're thinking about evolution, if we change the actual unit of natural selection to consider these processes and functions that are keeping life persisting, moving away from this kind of idea that's very focused on species that's defined by this particular, in this genetic way, to considering actually taking a more functional perspective and saying, if we can accomplish that, if that can be done, that this can be considered a robust unit of selection. In that way, we can actually put this into this evolutionary framework and make it, apply this way of thinking. But it requires changing that fundamental point at the basis. I see you nodding a lot, Stefan. I don't know if you have maybe something to add here.
Oh, I don't know. Although there still is the metaphor of selection involved in that, right? I mean, there's the question of whether Darwinian theories, notions of descent with modification is just sort of a subset of what's going on in the Gaian thing, right? So that there could be like little eddies or whirlpools of Darwinian things that don't necessarily then, are not necessarily coextensive with every possible thing that Gaian biogeochemical dynamics get after. I mean, I feel like, Lynn Margulis's critique of Darwinism was that kind of relentless notion of competition, and then also the unidirectionality of transformation in this kind of phylogenetic lineage, rather than the kind of more scrambled rhizomatic sort of thing that she, with symbiogenesis, wanted to point to. So I'm nodding in lots of different ways all at the same time.
Okay. Yeah.
Well, thank you so much. Dear audience, I haven't forgot your question, and I wanted to stress the fact that the next panel will start only at 6:30, so we actually have another 15 minutes. So I just decide that we can go over 6 p.m. I hope all my speakers here in the panel are okay with that. You don't have to run off. There are questions from outside. I'd like to put them in here at this point. Later on, I don't want to close the panel without discussing the ethics. I think this is an amazing chapter and connected to the conundrum of the Anthropocene, and maybe I'd like to later on come to it. Now I look at the questions from outside, and maybe I can launch the question to the specific person here, and maybe you can in a compact way answer. I'd like to start with a question from Sarah Amanutz. Nicole, any thoughts on altered implications of care, usually embodied, present, intimate, for your practice, with circumstances preventing your direct creation involvement with the Berlin microbes? And then she adds, by usually I mean in your descriptions of the care you provide under normal circumstances with the work.
Yeah, absolutely. It's been certainly a new challenge in terms of how I do think about my relationship with the work. This sort of remote manner of kind of putting the labor of care onto the wonderful team at Art Lab Berlin, which I'm very grateful for. And I think it's more of kind of like handing off that sort of labor or effort to someone else. So although I certainly still feel there's a relationship between me and the work and in its conception and sending the prisms over and consulting on how they should be cared for, I'm not actually doing that embodied caring for. So it is, I think, a different relationship that I would have then in terms of care with those particular prisms or this particular artwork, where I'm kind of, again, relinquishing some of the labor of care onto someone else. Yeah.
Thank you, Nicole. And there is a question to India from Ahmed Havadallah. Thanks everyone for the input. I would love to hear more from India on Darwinizing Gaia. Well, okay, we have that already from Sonia, and the tension that exists between these theories and the efforts made to reconcile them. I put this question to India, but then I actually like to take the other question into consideration for all of you. I think this fits perfectly. Here is Franziska Dahlmeier, who says, thanks so much for these inspiring presentations. I would like to know how the speakers place the artificial settings they researched or helped create within the wider scope of the Anthropocene. And then she asks, and maybe this can be also for all of us after India will refer to the first question, can they help the unbecoming of the Anthropocene? Maybe India.
Yeah, yeah, it is a big question. I mean, in responding to Sonia, I started to open this up again, but it really has to do quite a lot with tension in the world of evolutionary biology, and also of yeah, I mean, these ideas of competition, cooperation on Margulis and Dawkins, and there is a lot of tension there. And this paper from Doolittle, it's not very long, but it's quite thought-provoking, and it's just one means of attempting to kind of reconcile some of this tension by considering that the sum of all of these various biogeochemical, biological processes that are occurring on Earth are the thing that are pushing this homeostasis that is allowing life to continue to persist on Earth. and that if we change the way that we're thinking about selection units, then we can understand the way that there is this homeostasis that's persisting through time, because processes that are making Earth less supportive of life are going to not be as persistent as those that allow Earth to continue to support life more. I hope that that made some kind of sense. Perhaps I can hop over and post a link to this paper in the YouTube channel for people who are interested to read more.
Absolutely. Thank you, India. And I actually would like to come to you, Sonia. First of all, if you wanted to make a comment on the Darwinian Gaia, because I know in your theoretical thinking you also refer to Gaia and to the Anthropocene, and actually the ruins of the museum is the subtitle in your text and of your project. And maybe taken into consideration the challenges of the Anthropocene or Tulucene, we can also switch terms if we want, to speak in Haraway rhetorics. Do you want to refer to your project For the Love of Corals, or the Coral Project? And I remember in our interesting conversations, you also raised the ethic issue, talking about genetic modification, etc.
Yeah, I'll try to be quite succinct with that, because it's also a big question. But I think the idea that I'm trying to portray in the film also is this imagination of consequences. The idea of an ongoingness, of a constant unraveling, as opposed to finding a resolution or a solution. So yeah, I guess with Project Coral, the endeavor might give struggling corals the possibility to generate worlds of their own. But it could also be an endeavor of re-engineering corals, which may have the effect of rendering them reproducible, scalable, saleable, and kind of cut the world around them. So I think, yeah, one thing that is important for me, and that also linked to the other question you had earlier about scientific and artistic, is to think about the unintended consequences of certain projects, of things that appeared as solution. Yeah, and hold together those consequences at the same time as inheriting the ongoing devastation of our time.
Would you like to comment on that, Stefan, maybe?
I mean, I'm not sure. The ongoing devastation of our time is difficult to get, it's difficult to grapple with. Maybe, I mean, that question that you asked, Regine, about models might be a way in. That part of the practices that I take it that the people in Horniman are doing is, you know, this modeling is a kind of care, but I know that we might have a lot of, not skepticism, but caution about thinking about whether, you know, remaking Australia in the basement in London is, I mean, I guess that's one thing that's super interesting about the project, Sonia, is just that that work that they're doing to, you know, in the kind of in the ruins, or I guess it's not the ruins, in the context of ongoing colonial museum work, they're trying to kind of rescue in some way from, you know, this British imperial infrastructure, some kind of small mode of care, right? Which then of course kind of cycles over to this other British colonial enterprise called Australia. And, you know, trying to find hope within that is a vexed sort of thing. I don't know if that speaks to it really at all. I think I was just destroyed by the devastation of our time comment, which is pretty palpable right now. Yeah.
It is actually, it makes us speechless. And I'm very grateful, Stefan, that you at the beginning and in the end of your interesting video talk brought in the current pandemic. I guess it would probably crash the temporal possibilities of our panel way more than 2 hours if we wanted to elaborate our artistic and scientific and philosophical and biological and anthroposophical methodologies in connection to the current ongoing pandemic. Edda, actually, also in parentheses, I would have actually loved really to drink coffee with you here and to meet in the theater hall and to talk and even actually to get drunk and actually also to be able to smell one another and etc. End of parentheses. But yes, in times of destroyed planetary, one might get emotional. End of parentheses. But actually, coming back to the question of unbecoming the Anthropocene and my ideas and interests into the ethics. If you allow me maybe to make a little round again to reflect on that, and maybe Nicole or Tarah and India want to catch my ball. I see various points with the artistic research practices with Sonia, with Nicole and with Tarah, where we could come back to Puig de la Bella Casa, what is care? So we have this triplet, we have the labor work, we have the affections, and we have ethics and politics. And I remember in this interesting talk with India and Nicole that we also came very quickly, not only at the end of the conversation, to think about the critical impacts of care, especially of us humans wanting to do the care, either erecting Vinogradsky columns to get a better understanding of what microorganisms are, or actually to put the hands in the mesocosm and wanting to help the corals to grow, or actually think on the molecular level how animals and plants could come together and to erect biochemical installation. And apart from the fact, and this is also the conundrum, which is probably a basic conundrum for all of us, is when we want to think outside the human complexities, we cannot, because it starts already in the linguistic conundrum. We are here and we speak and we use this language, we use this alphabet, and it's actually, we wanted to do animal talk, we wanted to maybe talk in the biological languages. So it is, if only, if it's always a stumbling. But what actually comes to your mind when we think at that stage, a thought of unmaking of care, or maybe radically leaving the human territory and the possibilities of ethics?
I mean, what first comes to mind is certainly, I think for a lot of maybe individuals considering digging up some mud and placing it into, you know, a Winogradsky column, a prism, as I did, that wouldn't necessarily be an ethical conundrum, I think, for a lot of people. But as, or maybe it would, but perhaps I'm making assumptions. As I think a practice-based researcher doing this act of removing microbial life from, you know, where they had their established community and placing them into a prism where, yeah, some of them will flourish and some will die, and it's going to become this, you know, human created environment where I have to think about that violence, right, as an ethical consideration. And I do think it comes back to sort of staying with the trouble, as Donna Haraway suggests. And also, as Anna Singh talks about disturbance in her book, The Mushroom at the End of the World, and where's the disturbance too much, right? Where is that cutoff point? And I think for me, the fact that the microbial life, although very in an altered environment, is still capable of both living and dying, and also has that presence in order to hopefully prompt much more care, elaborating beyond just the experience of the artwork or my care to the artwork, that viewers will hopefully expand upon this understanding of liveliness to then care for and about our ecology much more broadly.
Who wants also to comment on that? So I'm just seeing, is there more questions from outside? I think there's no question from outside yet, although we have so many questions left. And actually also what we all agree on definitely is we get out of here always with more questions that we have got in, but this is actually good, that keep us thriving and critical and following research and observing. So I talked about cross communication. It is nearly time, but just still at the end, I wanted to, as I announced it, I wanted to give, for the last few minutes of our panel modes of care, I wanted to give you the chance. Do you want to cross question some of the speakers here, or do you have any cross commentaries here? Question to all of my 5 speakers. I guess this is just a start, and I think the dialogue will actually unroll, I hope, in the next few days, weeks, and months, and years. I'm very optimistic. So actually coming to the end of the talk, it was a very long panel, and we will continue at 6:30. At that point, I really, from the bottom of my heart, I want to thank you all, 5 speakers. Thank you, Sonia. Thank you, Stefan. Thank you, Nicole, thank you, Tarah, thank you, India, for sharing your interesting knowledge and ideas with us. I learned so much, and I would like to invite you all, and especially I would like to invite the audience to come and visit Art Laboratory Berlin in the exhibition. It is open until the 4th of October. Unfortunately, we cannot prolong the exhibition. Please bring at least 90 minutes along with you. And this already has to do with the 2-channel video installation of Sonia Levy, for instance, which is 25 minutes. And all the other artworks are also very temporal based. Plus, dear audience, you are also invited in the exhibition spaces. We have 2 venues to study the so-called resource books, where we share with the public articles, papers, and background information that is very much part of the artistic research that we're involved in. So thank you very much. We go into the break now. We have a little pause for the YouTube live stream, and then we all meet again live at 6:30 with our final panel with Ibn Kirksey and a final discussion. Thank you so much.